So I'm going to talk about something slightly different. I'm going to talk about, the title of my talk is Mathematical Innovation for Drug Discovery. There is mathematics that underlies what I do and what I'm going to talk about in this talk, but it's quite a bit less careful, I would say, than the mathematics that we've heard so far today. So my name is Lucy Colwell. I'm on the faculty here in the chemistry department. I'm also currently on secondment at Google Research. And so this is a picture of the kind of computers that I have never gotten to see in real life, right? They don't let me anywhere near these data centers, but we do get to use these computers virtually, even though. And in fact, Google actually makes them also available for academics to use. There is some kind of program, but you will never get to see them in real life. Like they're very secure. But they do allow us to build big models, which is kind of exciting. I guess I'm very interested in trying to make those models a little bit more careful, but we haven't got there yet. So what I'm going to talk about today involves hammers, essentially. So I'm going to talk about a project that involves, or is aimed towards helping with gene therapy. So gene therapy is a sort of emerging technology that can now treat and cure chronic genetic diseases. So there are FDA-approved therapies on the market that involve gene therapy. It's a very new technology, and there are lots of sort of concerns about it, but I think it is nonetheless interesting. And for me, this is very much a sort of demonstration piece of work. So it involves gene therapy, but it's something that you could do with proteins in general. Particularly, I'm going to talk about using a vector called the adeno-associated virus and sort of changing that vector. So there are sort of two main forms of gene therapy. There's germline and somatic gene therapy. So somatic gene therapy is where you change somebody you change their somatic cells rather than making a mutation in their germline cells. And so AAV is used in somatic gene therapy. It's in vivo, so you inject it into somebody, and it is a virus. So adeno-associated virus is a virus that exists naturally in the wild. We're all exposed to it constantly, so there's AAV floating around everywhere. And in general, it doesn't really cause you to experience an immune response, right? Because you're exposed to it all the time, you don't generally respond to it, and so that's sort of a point in its favor. And so here on the right-hand side of the slide are some different viral capsids or viruses that are used in gene therapy, and here is AAV, or adeno-associated virus. So this is adenovirus itself, which is a much larger virus. Adeno-associated virus is very small. It means you can only put about 4.5 kb in as the cargo in this sort of expression cassette. Why do we need a better AAV? Well, essentially, we do need the capsids that contain the therapy to get past your immune system. If your body rejects them and destroys them, that's not much good in terms of a therapy. And also, you need to be able to localize your AAV capsid to a particular cell type. So it'd be great if you could really address it. So you give somebody an injection, and the gene therapy goes to a particular part of the body where it's needed. And this is currently very, very challenging. This hasn't really been conquered. But in this talk, I'll actually talk about a much simpler phenotype. So these are sort of the two long-term goals of any gene therapy project. I'm going to talk about a much simpler phenotype, which is simply making a sort of integral virus. So this is a map, or a slide that explains something about AAV structure and function. This is a genome map here on the top of the slide. This is the whole genome of AAV. So it has these two main coding regions, this rep gene and the cap gene. So the cap gene is the one we're going to look at in a lot of detail. It produces 3 proteins that are called VP1, VP2, and VP3. So they get read from the single gene, and as you can see, they overlap. So they have a lot of shared region, and they have slightly different N-terminal pieces of protein. And so this cap gene is 735 amino acids long. It's a big protein, and we're going to think about it as producing just one protein. Right? There are 3 variants, but they're very, very similar. And so essentially it produces this one protein, and there's a 3D structure here of that protein, and 60 copies of that protein tiled together to form this beautiful, highly symmetric AAV capsid. Okay? And the way this virus works is that capsid contains the genome, and so the capsid gets released into a cell, and when it makes its way into the nucleus, so this is the boundary here between the nucleus and the cytoplasm of the cell, it actually it's uncoded and releases the genome, which then replicates And when the genome replicates, it produces more of these cap proteins, which then assemble a capsid around the new copies. So this is sort of the virus entering the nucleus. The genome is uncoded and released, and it gets replicated, and then you get many sort of child viruses, like child copies of the virus, each with their own capsid. So you can see that this is a kind of a complicated protein. It has lots of protrusions. It's a sort of odd shape, and they interdigitate together copies of this protein to make this capsid. So it's a fairly complicated process. This is something that would be really difficult to simulate using molecular dynamics or any kind of first principle simulation approach, just because it's such a large system. Right? 735 amino acids is already pretty big, and then once you have 60 copies of that protein, this capsid is huge. Right? It's like 25 nanometers across. This is really quite difficult to simulate, and so we're not going to perform any simulations here. What we are going to do is ask whether we can make changes in this cap gene, so changes in this protein that allow us to still form an integral capsid that packages its own genome, but nonetheless changes the protein itself. And the reason you might want to do this is that there are patches on the surface of this capsid that either interact with antibodies in the immune system or help localize a capsid to particular tissue types. And so if you can make changes in the surface of this capsid, then you can really change the properties of this virus. So what we're doing here is we're going to ask whether we can make a lot of changes in the protein that still allow this capsid to form, and so kind of give you a palette, if you like, of different variants of the gene that then might allow you to achieve these phenotypes that we talked about. So we're kind of making, this is an enabling technology. So packaging is literally whether the capsid variant makes it. So if I make changes in the surface of this capsid, I need to know if it's still capable of working. Right? Is it still capable of once it's replicated, forming an integral capsid that packages its genome? Okay, oops, sorry. And that's what this sort of little GIF is meant to illustrate. Many of the variants will simply fall apart and not work. So to do this, we're going to need to be able to experimentally test these variants. And this is where collaboration is key, because I am not a molecular biologist, and my colleagues at Google are also not molecular biologists. And so we did this project in collaboration with the Church Lab at Harvard, which is a big synthetic biology lab. And so they basically have this technology where you can take a small piece of a protein and print, using something called an oligo, different variants of that gene. So in this case, between positions 561 and 588, we can literally print exactly the amino acid sequence that we want, and then integrate it into the gene of the whole protein, and then express the protein. And so we're going to start off by thinking about single changes, and actually we made these across the whole protein, and then we're going to think about how to combine those single changes into multi-mutants, and that'll be within specific tiles, and I'm going to focus on this particular tile 21 as part of the capsid. So this is just a demonstration that we can make progress within a particular tile, and the idea is we could then do that within any tile. There's nothing special about that starting point. And so essentially the way this works is you print the sequences you want on a chip, you then replace the native sequence using the little piece of DNA made using that chip, and so you clone it in to the native sequence, and then you express the plasmid containing that altered sequence in a bacteria. And so you have this what we call a library region of 30 to 50 amino acids, this tile 21 region in this particular part of the protein. You're going to make something between 10,000 and 100,000 variants per library, where you get to specify exactly the sequence that goes in each of those variants. And that's pretty unusual to be able to do that. The library sequence can really be anything that you want. And so then there's this packaging assay where you have many variants in a library. Each variant is made into a recombinant plasmid that's transformed into a cell. And you do this at a dilution so that you just get one variant per cell, right? Because you don't want cross-packaging between different variants. And so you can then sequence the library at this point to understand how much of each mutant or variant you started with. And then you carry out the assay, which is a round of viral replication, and then you can sequence at the end to ask how much of each mutant you end up with. And we're going to measure selection, which is just the ratio of the counts after to the counts before. We'll often actually look at a log of that ratio. And we did some analysis of the experimental noise. There's tons of noise, like in every single part of this experiment. There's noise everywhere. And actually I have a slide in a second that will show you that noise in more detail. But basically it means that there's like one bit of information in this experiment. So for each variant we can tell you whether it works or not. That's it. So I said we made a bunch of single variants to start with. A single variant is either a substitution where we change the amino acid. You can see here that it was D-E-E-E and now it's D-E-E-D. So we've made that single change there. Or we're going to allow single site insertions where you stick an extra amino acid in to the capsid to fit it in between. And that's structurally quite difficult to understand how that works. And so we were nervous about doing these insertions, but in the end they seem to have been quite useful. So we started off, our collaborators made all the single site insertions and substitutions across the entire protein. It's about 30,000 different variants. They made all of those variants and tested them. And so then we had this sort of initial data set that we could use to try and design alternative sequences that contain many mutations. And so there is a large variance in the data. So here in green, I've shown you the experimental outcomes, and this is fitness on the bottom, a log of fitness on this x-axis. And so you can see that these green ones are all wild type. That means they're all the same amino acid sequence, but there's this big spread in their reported values using this assay. Right? So that means there's a fair amount of noise. The grey shows actually all the single site changes. So there is signal in this data. There are values that are sort of outside of this green band, but we basically felt that there was sort of, these ones were probably broken and the grey ones here were probably as good as wild type. Right? We're being a little bit conservative, but that seems to be, roughly speaking, what's going on with the noise. And so essentially we're going to model this as a binary classification problem. It turns out that, well, I'll talk more about this data later on, but if you make random multimutants, then you get, it's very easy to break the protein, as you might expect, but you do get some that are still viable. On the other hand, if you design your multimutants carefully using that single site data, using just a very simple additive model, then you can do much better in terms of designing sequences that are viable and are far away from wild type. So what will be important on the x-axis here is, well, this is packaging fitness relative to wild type, but what I'll sort of focus on later is actually the sequence distance from wild type. So you want to get sequences that are far away. That will be the thing that becomes important. So here's the results of the single site experiment. These are substitutions shown as a heat map. Basically the blue things are broken and the white things are normal. So these are the positions in the tile on the y-axis and the 20 amino acids on the x-axis. So we've literally made all of those changes, and you can see that there are some regions of the tile here that are hard to change. So particularly these sites here are very hard to mutate. They really only want to be their original amino acid, whereas later on in the tile things get easier. And that makes sense because the first part of the tile is sort of structurally at an interface between the capsids, so it's quite complicated, whereas the second part of the tile is more surface exposed. And so the collaborators actually started off with this very simple model where you just combine the single site variants to make multiple ones. You sort of choose good single site variants and combine them, and you have this sort of additive hypothesis, and you can use that to choose multimutants using that data. And so they literally took all these single site substitutions, measured sort of close to the wild type, and they used this additive model to choose things that were far away from wild type. And so here's the outcome of that experiment. There's a number of mutations on the x-axis. This is how far you managed to get from wild type, and this is how sort of the percent viable for each of those bins. So you can see that 2 steps from wild type, it's actually pretty easy. There's like almost 90% viables here, and that you can actually see that as a ratio of the green to the purple. So these bars are showing the number of sequences that were tested, and that's on this other y-axis over here, and the ratio of these bars is what gives us this grey point. So close to wild type, you can do pretty well at finding viable sequences just by combining single variants in an additive model. But as you get further away, out to sort of 7 to 10 or 11 to 20 steps from wild type, it becomes much more difficult. And so what we wanted to ask is, can we learn better models that we can use to choose multiple mutants with multiple variants from the wild type? So the first thing we're going to do is some post-hoc validation. We sort of have this set of data that was designed using an additive model. We're going to ask, can we train machine learning models that will do a better job on this same data? So the sequences have been chosen using an additive model. We're just going to ask for those sequences, can we sort of raise this line, right? Can we increase the accuracy using a machine learning model? And so we're going to train our machine learning model, actually using a set of data that we asked our collaborator to collect, where they just totally randomly picked sequences at various distances from wild type. And they thought this was completely crazy, and it was completely crazy. You can see that only 10% of those sequences worked, but it gave us a very useful sampling of the space. But even though many things were broken, we knew that they were broken, whereas before we didn't know that they were broken, and that actually turns out to be really quite useful. So there's a set of 11,000 randomly mutated sequences. We're going to actually make things even harder and say if we just focus on the 3,000, there's these double mutants here, and we also have all the single mutants, there was 1,000 of them. So we make just those 3,000 data points, can we then extrapolate from that data set to predict viable mutants far from wild type? So we now have two training sets, one with just 3,000 data points, one with 11,000 data points, and we're going to use them to sort of screen this much larger set that our collaborators made using an additive model. And if we do that, then it turns out this was that line from before showing their model, that actually even with a logistic regression model, which is shown here in blue, we can actually already raise that line a bit. So logistic regression is still an additive model, right? It's a very simple model. It just means that we get to learn the coefficients that we attach to each site, rather than in the case here with this additive model where we just assumed the coefficients were roughly all the same. And it turns out we can even do better than that if we learn either a convolutional neural network model or a recurrent neural network model. And so those models, which are trained on those 11,000 random sequences, actually give us a bit more lift, particularly in this region here, where we have sort of between 5 and 10 mutations. I'd say everything is really sort of struggling down here with 11 to 20 mutations, but we can definitely get further out using a machine learning model. Actually, what turns out what's even more interesting is that if we train the CNN on just those 2,000 random double mutants, then we can still actually get that improvement over the additive model. So somehow, actually quite a small amount of data is allowing a neural network model to lift that line. And I thought that was sort of interesting, because we all hear about these neural network models being very data hungry, but in this case we actually just needed a bit of data to get that lift. So then of course the question is, it's all very well we can use these models to screen sequences that have already been chosen using a simpler model, but can we actually use them prospectively, right? So can we really use them to design new sequences that are far from the wild type starting point, without thinking about this additive model at all? And so sort of set up an experiment here in which we had 3 different training sets. So we're either going to have that set of random doubles I talked about, the sort of set of random multi-mutants, so all of these mutants here, and we're also going to use this design set as a training set. So this design set has an extra experimental step involved, because you've got to sort of 2 rounds to get here, whereas these were all just chosen at random, so there's only 1 round. But we thought it would also be interesting to compare. So there's very different amounts of data here, and actually we're going to structure it in such a way that the sets contain each other. So the random double mutants will be the smallest, but it's contained by the random multi-mutants, which is B here in this diagram, and then set C is actually the design mutants and the random ones. Put all the data in. We thought that would be like the best possible model. We just use all of our data. I'm going to build 3 different types of models. We'll build RNNs, that's the top row here, CNNs, it's convolutional neural networks, and logistic regression models, because if actually the simplest model could do really well, that'd be great, right? These models are much easier to understand and much easier to interpret. Everyone feels much more comfortable with them than with these sort of much more complicated neural network models, and so that would be really great. So the first thing we're going to do is we chose 1 billion sequences uniformly at random at each distance from wild type between 5 and 25 steps. So that is easy to sort of think of as an experiment, but it's actually computationally tricky to do. Right? You actually have to choose 1 billion sequences at each distance and realize them, write them down in a file, and then run them through the model, which is sort of a pain. And so you do inference all those sequences. There's something like 22 billion sequences in total, and you get all the scores and you sort of rank all of the sequences. So that was one place where these large computers came in handy, because they're quite good at all of these sort of operations. That's a very simple thing. We're just going to take random sequences and use the model to screen them. The second thing we wanted to do, and so here this is just showing that in more detail, and we literally take the top n sequences. We take the top n sequences from each distance bin. So there's 22 billion sequences, 1 billion from each bin. Each model takes its top, I think it was 100 sequences from each of those bins. But what we're really hoping these models are going to do, and this is a picture that I've actually stolen from a paper a long an old paper illustrating the fitness landscape, so the relationship between sequence, so this is genotype mutations shown here, and function or activity is measured by height. What we really want the machine learning model to do is to make this model, right? We want it to give us a model of the relationship between sequence and activity. And so if we have a random starting point, a sequence we've chosen at random, the model kind of likes, we'd like to then ask the model, can you make this sequence better? So can you sort of make changes to the sequence using the model in order to kind of walk up the gradient and find a high point? So we did that. Basically, we had all these sequences that we'd screened, randomly sampled. We used the model to improve the sequences in a really moronic way, just by making potential changes and scoring them using the model. And we just keep iterating round and round that loop to find sequences that the model likes better than the random starting points. And then we had this massive set of sequences that we asked our collaborators to test. There's basically something like 23,000 sequences for each of these 9 models. There's 3 model types, 3 training sets. That makes 9 different experiments, and we're going to give them a bunch of sequences that they just screened. There's 100 sequences at the top here that each model just screened, and then a bunch of sequences that they improved. There's lots of data. We actually really just thought none of this would work, and so we wanted to get some things that worked, so we did lots of sequences. So basically what happens is, here I'm showing just what happens with the screening of random sequences. So there's a sort of percent viable on the y-axis, and there's distance from wild type on the x-axis, and that additive model is shown here in grey. So actually, the additive model did pretty well at getting things that are far from wild type compared to these machine learning models. Interestingly, among these this is just the logistic regression model. There's 3 different training sets for that logistic regression model, and the one that sort of has done best, I think here is the green line, which is actually the one trained on the set of random data. So even though the designed set had all the random data and a whole bunch of extra design sequences, it somehow wasn't as good at finding sequences as the randomly trained model. So then, that's logistic regression. If we look at the neural network models, they're a bit more robust to the choice of training data than the logistic regression model is. Right? So here you can see that the curves look a bit better. I haven't done a very quantitative job of analysing this. They're basically doing better at finding sequences certainly close to wild type, and then they sort of drop off. But then what happens if we use the models to improve the sequences? So here you can see that actually things get a bit better. We're able to do significantly better than the additive model. We're able to find sequences quite far away from wild type. This is like 25 steps. Sorry, the x-axes are a bit small here, but it's a lot further. The additive model you get maybe 15 steps away, but with these models we can get maybe 25 steps away from wild type, so quite a bit further. Interestingly, the best model of all is the logistic regression model trained on the random data, which is just amazing. It just keeps going. It's incredible. What we can see, though, is actually that logistic regression model, this is showing the diversity of the sequences that it chose. So somehow here, these sites, there were a lot of different amino acids allowed, whereas other sites were very sort of conservative. There weren't that many changes. So even though the logistic regression model has got really far away from wild type, it hasn't found sequences that are all that different. It's found lots of sequences that are very similar to each other. Whereas if we compare that to the neural network models, you can see that these are finding sequences with more diversity. So this is just showing a measure of the information content at each site in the sequences. And you can see that here the positions are much more conserved, whereas here many more amino acids are sampled. And there's also some differences in if you look at these plots more generally, there's sort of these stripes in the RNN plot, which sort of says that the RNN shows residue-specific preferences. It has the same opinion about, this is valine here, this particular line, valine across lots of different sites. So you sort of see these stripes, whereas the logistic regression model, it's totally different. There's no sort of stripiness at all, and the convolutional model is somewhere in between. These are these plots that everyone makes. This is like a t-SNE projection of the data. It's like PCA, basically. It's trying to show you something about where the different models chose sequences. So we've used the sets of randomly chosen sequences to kind of make a framework for this projection, and then we've projected the sequences chosen by the different models onto it. I'm not really sure what these plots ever tell you, but you can see the logistic regression model plot is sort of more concentrated in particular areas of the space, whereas the other models are sort of a bit more all over the place. Yeah, and so basically what we did in this project was we've sort of built these machine learning models to find new protein sequences, and they actually did pretty well at finding protein sequences that worked. And we sort of validated these predictions experimentally. We were able to find sequences that were really distant from wild type, and the sort of more complicated models tended to be able to select very different viable sequences. And I guess the conclusion is that randomly drawn training data was much more valuable than sequences that were chosen rationally. And that was very surprising. And I guess I'm trying to sort of follow that up with some other collaborators right now, because how you choose your training data is a really important sort of decision about how you set up an experiment, and this is a very surprising conclusion. So I don't know how broadly applicable this conclusion will be. We certainly didn't expect it to be true in this case. We also found that both simple, by which I mean logistic regression, and more complicated models were useful, perhaps useful in different ways. So with that, thank you very much for listening. I'm more happy to take any questions, but I know it's also lunch next, so we could also just talk over lunch. Thanks.
Just one question, fabulous talk. Out of curiosity, obviously, mutations happen by chance. Happening one mutation is fine. Two, sometimes happens. Three, not very often, and so on and so forth. How likely it is that the wild type sequence is optimal according to your model?
Yeah, I'm sure it's not optimal. It depends what you're looking for, of course.
It does.
Right? And in terms of maybe it's very robust to, because as you say, single mutations occur quite often at random, right? So it could be the wild type sequence, although it could be better at directing to a particular tissue type or whatever it is you're selecting for. It could be very robust to those single changes. But yeah, there's a huge sequence space to explore, and evolution has clearly explored part of it, but there's just this frontier. Yeah. So
the wisdom from protein engineering is you get what you select for.
Yes, exactly. Yeah, no. So I think that's key, right? We're always going to get what we select for, but here I think what we're trying to do is just change the way in which you carry out that search, right? So carrying out that search either with like a randomly generated library, a huge random library, or with sort of people make these libraries where you allow certain changes at certain sites and there's still randomness. Here we're trying to ask if you can sort of search in a different way. Okay. Yeah. Thanks. Yeah, please.
So when you showed your diagram of your kind of area that you want to explore,
yeah,
two two maxima, one a global
right,
maximum and a local maximum, and you're winding up there.
Yeah.
When you did your single mutations though, you had lots of dark blue at the top, but you had one pink, which was slightly better, and then it's surrounded by lots of quite light blue. Surely these global, the local maxima are all going to be really sharp.
I think that's a really interesting question. So I'm sort of involved in a project right now where we're simulating these landscapes, right? Because you can just ask, if I simulate these models, sort of landscapes using different models where I control like the sharpness of the peaks or how many peaks there are, how far apart they are. Then how do different models work at sort of exploring that landscape? Because I think that's a really good point. They could be, it's like activity cliffs in chemistry. They could just be really sharp, discontinuous. Yeah. I kind of hope that maybe that isn't, because that sounds pathological. I hope that kind of isn't the case, but it could be. Well, yeah, right. Yeah, there must be some. Oh, yeah, go on.
So in terms of computation complexity, so your neural network comes up with a prediction, and then you'll probably have to validate it, and then make sure the packaging is right. So what was the accuracy is the first question I have in terms of when it was coming up with new sequences, how accurate they were. And is my gut feeling is that the model which is going to check the stability is going to be more complex.
Oh, it's not a model. So we don't have any models for checking the stability. It's an experiment.
Okay. Oh,
yep.
You've not done that bit.
Yeah, there's no modeling. Like I didn't do any MD, anything. Literally, we just propose the sequences, the models choose the sequences, and then we test them.
Okay. Now the testing phase.
Yes.
How computationally?
There's no. It's not. It's just an experiment. Yeah. So that's why I said at the start, like everyone, people have these really complicated and beautiful theoretical formulations, and it's just that this is too big, this system, I think, to fit. Yeah, but it still was computationally intense, right? Like being able to screen all of these sequences. The models are fast. Like this kind of model on a single GPU can screen, like, I would say 250 sequences per second. So it's fast, but it's still, if you're trying to screen billions of sequences, that still adds up. Yeah, so, yeah.
A small question.
Yeah.
But, your data was, the variability was primarily at the level of a gene, right? But say if one would like to also include the role of the environment, right? So include also external variables as opposed to on top of the variability.
Yeah, absolutely.
What would be your comment on,
So you can do this, and actually I'm sort of involved in a collaboration right now where we're actually not changing the sequence, but we're changing the environment, right? So like the pH, all the sort of growth, like you have a vat of stuff and you want to, like you make a sourdough bread starter, right? And you want it to grow, right? How should you? And so we're basically using similar modeling approaches to try. It's like you're searching a landscape, but the landscape, instead of being in sequence space, is in the space of experimental parameters. So I think you can use similar approaches, but it just gets very hard because you have there's many factors. You can have like many different temperatures, many different pH levels, many nutrients, many lighting conditions. It just kind of goes on and gets big quickly. So sequence space is large too. I don't know, if we could use machine learning models to actually search large spaces, it would be really useful. But I don't know how to do it yet. Yeah. Okay. Thanks.